Good afternoon, and welcome everyone to the Verspeeten Clinical Genome Center virtual tour here at London Health Sciences Center. The Verspeeten Clinical Genome Center is the first of its kind in Canada, and so we are delighted to have you all joining us this afternoon for this exclusive behind-the-scenes look at all of the incredible work that's happening and how it's helping to change patient care. My name is Pam Taylor, and I am the Director of Development at London Health Sciences Foundation. Together with my colleagues and the amazing staff in the Verspeeten Genome Center, we are so happy to have you joining us and so grateful that you are here. Thank you to Archie and Irene Verspeeten and to all of our donors. You make all of what you are about to see and hear possible. A few things about today's tour. Everybody is currently muted, and that is to protect with the sound during the presentations. We are recording. And last, at any time this afternoon, if you have a question, please feel free to put it in the Q&A chat box at the bottom of your screen, and we will do our best to answer all of the questions at the end of the presentation. Again, thank you so very much for joining us, and without further ado, we will begin. Our first presenter this afternoon is Dr. Beckham Sidikovic. Dr. Sidikovic is a professor and program head of the Molecular Diagnostics Program and the Department of Pathology and Laboratory Medicine. Dr. Sidikovic's research interests involve application of genomics technologies to clinical diagnostics, with particular focus on development of genomic and epigenomic biomarkers in hereditary and acquired genomic conditions. Dr. Sidikovic serves as the clinical and scientific director of the Verspeeten Clinical Genome Center and Translational Genomics Facility, which focuses on development and clinical implementation of genomic technologies and biomarkers in the care of patients with genomic disorders, including hereditary genetic conditions and cancer. The center utilizes advanced bioinformatic approaches, including machine learning on patients' clinical and genomics data, to develop novel diagnostic tools and technologies. Dr. Sidikovic will be speaking to us today about genomics and technology, and he is broadcasting from the Verspeeten Clinical Genome Center labs.
Well, hello, everyone. Thank you, Pam, for that very kind introduction. So I am the lab guy. I figure I could spend the next 10, 15 minutes telling you all kinds of things about lab things, but instead I decided it'd be a lot more fun to do a tour right through the lab. I have my iPhone here. I'm going to hold it in my hand. And in advance, I apologize for any occasional mishaps that might happen. I told the lab I'll be walking through so they don't think it's a strange thing that I'm walking around with a camera talking to myself. So they know that there's a tour going on. But before I do that, I'd really like to, thank everyone. Joe and Pam's thanks. Your support plays a huge role. It does impact families every day. You have a really good roster of speakers. They're going to tell you the scope and the breadth of the impact that this is already having. With the generous support from Verspeeten family, we've already been able to start some really, really interesting initiatives you can hear about. We are actively helping diagnose and change management and impact positively the management of patients with cancer. As part of this initiative, we're doing some breakthrough genomic and epigenomic research that's creating ripple effect regionally here, locally, provincially, having impact in changing the practice guidelines across the province, and truly making some innovation that is world-class with some of the epigenomic work. And you'll see at the back end of my tour here in the data analysis room with some of our bioinformaticians discussing, we are truly leading some efforts that are international scale where we are at the N of 1. So I guess without the further ado, I'm very excited to have an opportunity to grab my phone and walk to the lab with a couple of people that are going to be joining me. So bear with me just for one sec as I gear up and flip my camera here. Hopefully it works. Okay, there we go. So right now in my office, you can see a nice view down. Here's the cancer center. We have the Victoria Hospital here on the 10th floor. We're locked out. We're not in the basement like most of the other labs that I've ever worked in. So as I walk out here, here's Sue. She's an administrative assistant here, and she takes care of our patients and phone calls and so on as we get them. As I walk out here, I see these two lovely professionals dressed in lab coats as they should be. So this is our sign, the Verspeeten Clinical Genome Center. We had a really nice opening ceremony with Archie present. This was almost a year ago now. But before we get started with the tour, I'm going to do a quick introduction here. So this is Jen Kirchhoff. She's our senior technologist, and she manages a lot of the patient sample workload and processing of these patient samples as they make their way to the lab. And here we have Haley McConkey. She's a research coordinator from the Archie Verspeeten Genome Center, and she is also our most recent graduate student. So she's doing her PhD as we go along. So they are going to be taking us through the labs and telling us all about it. So let's get started. I will just follow along. So Jen, where are we going?
Yeah, so we'll start through our molecular genetic lab. First testing or first step is our sample receiving, where we then take the DNA out of the samples. So we'll come on through here.
All right, so we're getting into our accessioning and DNA room. I'll just do a quick spin around here for everybody to see what this is. You can see a whole bunch of people and a bunch of equipment. And Jen, what do we do here?
Yeah, so this is where we receive our samples. We receive multiple different types. So anything from peripheral blood to tissue to urine to paraffin embedded tissues. We then process that and take the DNA out. So our techs that are in here now are working through collecting the DNA from tissue samples. Heather in the corner is taking it from FFPE.
Next up,
Slide for us and show us the type of sample or the tissue that we want to process. Vanessa here is preparing samples for oncology testing that we will run through and look at the genetic profile on tumor specimens.
Excellent.
And Alan here is processing some blood samples through our system.
Alan, how many samples do we get through here in a year?
In a year, roughly 15 to 20,000.
That's a lot of samples. So these are all individual DNA samples from patients coming from different tissues that we're ultimately are going to have to analyze. And before we analyze these DNA samples, I guess we're going to find out what happens next.
Yes, come on through here.
All righty. We did not choreograph this. We're completely ad-libbing, just so you know. I know it looks professional.
So now we're coming through a corridor where we start to prepare the DNA specimens. We've now taken the DNA out of the sample, and we want to prepare it for the eventual analysis. What this means, sometimes we make many copies of the DNA so that we can see it through fluorescence or through tagging. We have different types of rooms where we can put DNA and we can't put DNA. Coming through. So here in this room, we've got a liquid handler that will do some of the manual preparation for us.
Essentially, we got robots that can do a lot of the pipetting that we don't have to do by hand. Okay.
Yes.
All right. So once we prep the DNA samples have to go to other equipment.
Yes. And so once the sample is prepped, we then bring it into this room where we can actually make multiple copies of it. We can look at the different weight, and the different weight will tell us what the actual underlying genotype or DNA is. That's what this large instrument here does for us. It will look at the weight of the sample, which will tell us what the patient's DNA.
Okay, so what Jen is saying is we have machines that can measure the weight of the DNA molecule, which is minuscule. And if a patient carries a mutation, this DNA weighs different amount of weight, and we can tell by just running it through this machine and find out if a particular patient has a mutation. And in general, that can tell us how better to diagnose the patient or give them access to targeted therapy. Let's move on.
In this room, we have
Hello, everyone. Hi.
Technologists hard at work analyzing DNA. So you just saw Irene and Emma there. They're both working through analyzing tracings of DNA, which again will tell us if a patient has a mutation or not. down at this hall, we have Cassie and Stephanie, both involved in some of our translational work. So Cassie there is looking at NGS sequencing, all those red, blue, green, and black letters tell us if there's a mutation in the patient.
What are the pretty pictures here? Okay, there we go. Steph, you want to tell us what these clusters mean?
Sure. So we're working on looking at some methylation syndromes. And so we analyze data that kind of generates this plot that you're looking at. And so our red dots represent positive cases that have proven to show to have a methylation signature, and are positive, whereas green is our control group. So if we test a patient and that patient is plotting with our positive cases, we can diagnose them with a methylation signature.
That's awesome. So in kind of, you're going to hear a lot more about it from our bioinformaticists. But this is the one thing that we're the only people in the world that do. So we can tell based on patients' epigenetic profiles whether or not they fall into a cluster of patients that have a disease versus ones that don't have a disease. And that's some really exciting work that's ongoing. Thank you, Steph.
Thanks, Steph.
I guess we're going to go off to one or a couple more rooms before we get to the data analysis room.
Yes.
Oh, what's going on in here?
A quick little stop at this room here. It is our.
Hello, guys.
Hello. So these are all our genome analysts in here. Their role is to really look at variants and interpret if they are pathogenic or disease causing. Everybody has lots of variation in their DNA, and so we see it all the time. These guys tell us if it's causing disease or not.
And all our graduate students, postdocs, bioinformaticists, PhD level staff that are at the same time also doing a lot of the research. Erin over there in the back corner is our most recent hire into the center. She has 2 PhDs. I think it's very impressive. Okay, see you guys.
Thank you. And then this is our
oh, we got some pretty equipment here.
yeah, so this is some liquid handling equipment for us. So essentially robots that will help us pipette and prepare samples for sequencing so we can see the type of or order of DNA sequences. In the next bay here, we have Kara and Sherry. Kara is working through some NGS library prep while Sherry is doing some sequencing analysis.
Sweet. And we got a lot more people here hard at work. And notably, they're all wearing lab coats, and that's a good thing.
Yeah, so in the corner, we have Joanne and Alita. They're both working through some sequencing analysis as well. Joanne there is looking at the structure of genes so that we can compare and see different sequence variants.
And just so you all know, I mean, we all got DNA in our blood. In order for us to know what this DNA does, we have to go through really, really fancy processes and equipment. And ultimately, to be able to resolve what each one of these letters means. We have 6 billion letters. Only changing one single letter may ultimately result in a disease, may ultimately result in whether or not a patient gets targeted cancer therapy or not. So this entire process we showed so far is all parts of the process to answering the particular question. So when a patient sees a doctor and the doctor tells them that they carry a mutation that's going to give them access to a targeted therapy, a lot of this work happens in the background every single day on tens of thousands of patients in any given year.
And I'll just point out, because we don't want to leave out Mackenzie and Jasmine, but they're also going through, Jasmine right now is our most recent student. So Mackenzie is training her. So we are constantly training new students and performing education as well through here.
Hey guys.
Hi. And then these are our high power instruments that are up and running 24/7, generating terabytes of data. They're all sequencing instruments. So telling us order of nucleotides and will be helping us look for mutations in that DNA sequence.
I'm going to simplify this a little bit, okay? So back in the day, it took forever to sequence the human genome. And not really forever, years, 20 countries, many labs, billions of dollars. Now we can sequence a human genome on one of these instruments in 24 hours. Just because we can sequence it, it doesn't mean we can interpret it. So that's where a lot of this personnel, knowledge, experience, data analysis kind of comes in play. Okay, let's move on. So now we're going to make our way to the data analysis room where our bioinformaticists are working hard. And I think as we go down that way, Haley, maybe you can tell us a little bit about what our data analysts do every day. You are a more recent PhD student.
Yes.
You do a bit of that work yourself.
Yes. So our awesome clinical team generates all of our data, and what we're looking at mostly.
Hold just one thought for a sec. We're going to wave here to Dr. Holland. He's our section of molecular genetics, working hard at what?
I also do a value assessment and case report signing out.
As you can see, Dr. Lin here is reviewing manuscripts and papers and trying to figure out, what's happening with a particular genetic mutation. Okay, see ya. Okay, sorry, Haley, didn't mean to cut you off.
No,
Yeah, so tell us a little.
Okay, so what I was saying was that our awesome clinical team generates our data for us, and then the analysts in the room that we're about to head to will do the bioinformatic analysis to understand methylation in particular. So we're looking at using epigenetic data to generate signatures for disease. So that ties back to what Steph was talking about. And we are often trying to map new episignatures, which are the epigenetic biomarkers, to new syndromes, a lot of rare disease, but we're also moving this into cancer to see if we can help classify and diagnose cancers and maybe understand better what treatments a patient might be able to use based on their methylation data.
So am I opening it when I say that we're the only place in the world that can use epigenomic data to...
No,
okay,
I don't think
fair
so.
enough.
I
All right.
think other people are doing it, but not at the level and not with the same amount of data we have in our database. So our database is really what sets us apart.
Fantastic. So as we walk into this room, I should mention a lot of this work has sparked some really interesting collaborations internationally. We currently have a large network of clinical sites, upwards 200 different hospitals that are participating in this research, and multiple laboratories around the world that are adopting these technologies for diagnosing patients.
Exactly.
So
this is our bioinformatics room here, and we have many different team members in here, ranging from postdocs, grad students to bioinformaticists. So Risa on this side is one of our bioinformaticists. Mike is also a bioinformaticist. And then we have Sergey over here, who is one of our postdocs.
okay, well, thank you very much. Now, I will thank both of you for this wonderful tour you've given us. And I'm going to just flip the video here for one sec. And I'm going to give everybody here a wave. So now you're going to hear a little bit more from Pam before Risa and Mike take on. Thanks very much.
So thank you, Beckham, Haley, Jennifer, and everybody along the way. That was fantastic, and how interesting to be able to go behind the scenes into the labs. Our next presenters are Risa Relator and Mike Levy. Risa is a clinical bioinformatician at the Pathology and Laboratory Medicine Department. Her role includes software development for research and clinical use, pipeline and database maintenance and update, as well as data analysis. Mike is a bioinformatician at the Verspeeten Clinical Genome Center, where he integrates computational approaches to improve efficiency and help develop new diagnostic tests. He works on the EpiSign project, involved in the development of analysis pipelines and biomarker discovery for the diagnosis of rare neurodevelopmental syndromes, and works with the clinical diagnostic team on development and maintenance of their clinical bioinformatic tools and workflows. Risa and Mike will be speaking to bioinformatics, and as you can see, they are broadcasting from their offices.
Hello, good afternoon to all of you. It's so exciting to be here. You may have heard what goes on in the labs, and they kept on repeating terms like episignature and methylation. So I guess what Mike and I will be doing today is to give you an overview of what the work that happens after the data was generated for data analysis. So we will be talking about EpiSign. So this is a clinical diagnostic test that we're implementing in LHSC. And basically what we're doing here is a biomarker discovery So think of this as, for instance, you have two individuals. One is affected by a certain disorder or disease, and another one is unaffected. So what happens is we find differences in methylation. So methylation is a naturally occurring chemical process in our body, and we can actually quantify this. So there are many array kits that we can use to detect the methylation. And for some individuals, methylation levels in certain regions in the genome may be higher, and for some individuals they may be lower. So for instance, we have here methylation detected for these two individuals, where for the affected individuals, these regions here may be higher hypermethylated or higher methylation levels, and for the unaffected, they may be lower. Or for other regions, they may be higher for the unaffected individuals and lower for the affected individuals. So what we want to do is find such regions which we can make use as biomarkers in detecting or for diagnosing other patients. And these biomarkers are what we use in defining what we call the episignature. So the discovery project for EpiSign is actually a collaborative work. So we have major collaborators coming from across Canada and USA, and also the Netherlands, France, Italy, and Australia. And as our network expands, so we have now some collaborators coming from UK and as far as Japan. So to give you an overview of what we do for biomarker discovery, so first we establish a patient cohort for a certain disease or for a certain disorder, or in some cases the collaborators will reach out to us to request for analysis for a certain type of disease or a certain type of gene causing disease or mutation. And using in this patient cohort, samples are collected for these individuals, and the clinicians will also collect patient information that we will be needing for analysis. So this information such as age, sex, or like genotype information, or possibly phenotypic representations of these individuals. And as you may have seen in the lab tour, so after these samples were collected, so our very talented technicians will then generate the data, the methylation data, together with the patient data that we have collected. So after data generation, so for our data analysis, we actually use a pipeline which integrates some data mining and machine learning algorithms to find these biomarkers. So we use the data mining to determine patterns that are common in the affected individuals that are not present in the unaffected, so we can determine which regions we can make use as biomarkers. And the machine learning algorithms we use to train and build predictive models that we can later on use in the diagnostic as a diagnostic tool in testing other patients. And once we have established an episignature for that disease or syndrome, then we can now update that into our integrate that into our EpiSign knowledge database and update our clinical diagnostic list, so we can add that episignature in our list of diseases that we can test. So from conception up to present, so there have been many improvements in the EpiSign. Particularly, there have been many improvements in our pipeline that we use for the discovery. And also there have been several additions in terms of, for instance, the sample size or database size, and also the number of signatures that we have in our clinical test. So for instance, 2 years ago, we have like 28 signatures that we can test for, and now we have currently at 70 syndromes or conditions so we can test for. And our technicians have now been adding new samples at an average of 300 new samples a month. And in 2019, there were only like 1 and 2 analysts working on this cohorts, but now we have as many as 7 to 8 analysts. And we expect that as we continue to expand our network, we'll be also expanding our signature set. So extending into, for instance, we have environmental exposure related diseases or cancer, and we expect that we have more cohorts to come. So I'll be welcoming Mike to talk about how we use the biomarkers that we have discovered in the clinical setting.
Hello, everyone. I'm happy to be here. So thank you, Raisa, for kind of describing to us how we discover some of these biomarkers. And I'm going to talk about how we put this to use in the lab. So some of these steps you will have kind of seen as Beckham walked around the lab. So the first thing that happens for this analysis is that we get a blood sample from the patient. And some of these are from people here or in Ontario or Canada, and some of these are sent to us from all over around the world. The next step in the lab is isolating DNA from blood. We have some automated machines to help us with this process. The next step, once we have the DNA isolated, is to process it specifically for methylation analysis. And this includes things like treating the DNA with chemicals that will actually label the DNA methylation changes so that we can detect them. It actually labels them with chemicals that will fluoresce. We put the DNA on these little devices here. That's called a microarray. This is about the size of a credit card. And we put these in a machine that shines lasers on it, and it causes the DNA to fluoresce, and combining a microscope with a camera to actually detect those changes. Once we have detected those changes, then we get to the analysis stage. And we also add the samples to our database. And then the last step is the results after they've been analyzed. A report is issued back to the submitting physician from our clinical geneticists. So now I'm going to give you an example of how we would assess some samples from a patient. So in this example here, we see some results from two different patients. The first step is to compare the DNA from a patient to the DNA from patients who have all of the different syndromes that we can detect. So for example, for patient 1, each one of these dots represents what we call an MVP, or methylation variant pathogenicity score. It represents the probability that the patient may have one of these syndromes listed along the bottom here. And you can see that they have 0 score for all the different syndromes except for one syndrome that's called Soto syndrome. So that would tell us that this patient likely has this Soto syndrome. Patient 2, for example, which has low scores for all of them, would suggest that they may have a syndrome that we can't detect yet, which is why it's so important to keep adding to our database so we're able to detect more and more syndromes. So the next step, we use some additional algorithms to validate these results. We use what are called clustering algorithms. And a clustering algorithm will group samples together. It will group things together and more similar. So for example, on the left, we're using something called multi-dimensional scaling. And the green dots are represents samples from patients who don't have this Soto syndrome. And the blue are from people who we know have the Soto syndrome. And then depending on where the sample from the patient in question lands, we can determine whether they're likely to have this or not. We also look at this through something called a hierarchical clustering with a heat map. And you can see the same type of results where we have the samples from the two patients, which will be landing in one of the two sides to tell us whether or not they are likely to have the syndrome. This bottom part here is called a heat map. Each column here actually represents the DNA from one patient, and each row represents a single spot in the DNA. So we're able to analyze individual spots in the DNA and measure the DNA methylation very accurately. And if it's blue, that means they have low methylation at that particular location. And if it's yellow, it means they have high methylation. And we can see that for this syndrome, people who have the Soto syndrome have lower methylation at all of these locations that we examine. So to summarize the data process in the lab, as Raisa mentioned, the number of samples that we can process per month has gone up quite a bit in the last few years as we've been able to add more people to the team and identify more of these biomarkers. And as we have been processing more and more samples, we're able to add more samples to our database at a much higher rate. Because ultimately, the goal of adding more samples to the database is to be able to detect more syndromes. 3 years ago, in 2019, we released the first version of our EpiSign clinical test with version 1. And at that time, we could detect 28 different syndromes, whereas today, the current version that was released this year can detect 74 syndromes. And we are currently working on version 4, which we release next year, which we expect will add another 30 to 40 syndromes to our tests for what we can detect. Thank you very much, and I will turn it back to Pam.
Okay, thank you, Raisa and Mike, for that very informative presentation. Our next presenter this afternoon is going to be Dr. Mike Cador. Dr. Cador is the Chief Laboratory Officer for Pathology and Laboratory Medicine. Dr. Cador has led the transformation of laboratory operations, including a redesign of all core labs across the city, regionalization of services across Southwestern Ontario, and commercializing genetic diagnostics globally. Prior to joining LHSC, Mike worked extensively across the health and life sciences sectors in the United Kingdom, Europe, Middle East, and Asia, focusing on health system strategy and performance, innovation, and commercialization. He has worked with boards, executive teams, physician leaders, and policymakers in numerous organizations, including academic health networks, community and primary care organizations, universities, private medical insurers, pharmaceuticals, regulators, and local governments. Mike is a PhD engineer trained out of Oxford, United Kingdom, and Waterloo, Canada. Dr. Cador will be speaking to the Verspeeten Clinical Genome Center Vision, and he is broadcasting today from his office.
Thank you for the introduction, Pam. I am honored to be able to take the time to speak with the group here today. Oh, here we go. I think it's back on. I was appreciating the radio show, but we can do the video as well too. I'm just going to zoom back a little bit for the vision of the center as a whole. And no doubt, some of you are probably wowed and impressed walking through the science and the medicine, and perhaps equally, maybe equally confused, because it's a fairly dense subject matter, and we're, as you can probably guess, pretty excited to share this sort of information with you. So if I just step back a moment, and maybe you could kind of pick it up in my background. I come from a diverse career path of having been an academic myself, a researcher, as well as a consultant across a variety of life sciences industries, and have seen sort of all sides of how our health system works in a variety of different continents. Prior to being in life sciences, I worked extensively in a variety of other high-tech industries, non-life sciences oriented, and it always sort of struck me that when I got over to the life sciences side, particularly healthcare side, that this whole notion of R&D and your service delivery suddenly became two separate things. I'd worked in numerous organizations and had colleague engineers who've worked in high-tech companies where their R&D functions and their product delivery functions are in the same organization, and those there's a great handshake between them, and we get all sorts of wonderful new products and services out of these industries. But in healthcare, it's as though research has to be in a box on the side done by researchers, and then there's clinicians who are going to care for patients in the health system, the hospitals, who will do the care once all the research is sorted out. But these two, have a big barrier in between them. And in my role here now, leading our clinical laboratories in the city, that was a barrier that I'm hugely passionate about addressing. And you're watching and witnessing and walking through the Verspeeten Clinical Genome Center and starting to see what happens when the walls come down and research, development, and clinical care are happening together. And then another sort of personal passion, and you'll again, I think you probably experienced it from those who've presented already, is the role that the diagnosis and the diagnostics plays within clinical care, and how that can make a difference for each of us, on our own personal journeys with our own health and wellness, or our family members. You know, something as trivial as an issue with your car, the engine isn't working well, you take it to the shop, the first thing they do is they plug in that little cable underneath your driver's seat, and they read the codes and they tell you what's wrong with your car, and then they fix it. If they couldn't read the codes, if they can't tell what's wrong, then you can't fix the car. You can't even tell the driver of the car if their car is safe to continue on with, or if they should take it easy. They can't give you any information. So, you know, we know how important a diagnostic is for something as trivial as an automobile. It's sometimes easy to forget. And if those of us who've had firsthand experiences with cancer or family experiences with cancer, we know that getting an answer is so critical. Like, what is affecting me? Your physician team, your care team is equally trying to get that information so they can best determine the course of action to take with you. So having the privilege of driving this sort of work together from a diagnostic lab allows us not just to address what one kind of treatment could be or one sort of patient group might benefit from, but to provide diagnostic support to transform the care of a whole range of cancers, a whole range of even non-cancers, and allow care to progress and get moving. So the vision of the Verspeeten Clinical Genome Center is really that, to merge the worlds of research and development and clinical innovation in the space of diagnostics, so that we can start making more significant differences in what's possible from a care perspective. The other angle that I want to share with you, and there's some other great people that are going to talk a little bit about some of the specifics of what the lab is, how big we are, what we've accomplished. But I wanted to sort of put this frame on it around the business of academic healthcare. So that vision gets enabled by running a business properly. And I don't shy away from using that word in healthcare. I love using the word business in healthcare because we graciously take donors' money and taxpayers' money, and we have to put it to use in the best possible way. That's the business of academic medicine. And there is a typical sort of path of innovation from, you know, exploring an idea, developing an idea, and then translating the idea into the real world, maybe as an early pathfinder and then a sort of clinical mainstream. And what's very curious about most public healthcare systems, particularly ours in Ontario, is that the Ministry of Health and the other funders who you know, allocate the tax dollars to the health system for care, they live behind that big wall as it relates to R&D and clinical. So they will fund evidence-based care, which sort of makes sense. You don't expect them to fund things that they don't know are going to work or not. You expect them to use your tax dollars wisely. The issue, of course, is, well, how do we generate for them the evidence and the proof that these new emerging technologies are effective? So you have to manage that side of the business differently. And you can get money from public grants. You can get money from private grants like pharma companies and med tech. And that gets us so far, and that's a traditional model that works. When you add donor dollars into that mix, suddenly incredible things happen because we come with money to the table, and it unlocks all of those other groups digging deeper into their pockets. $1 becomes $5 or $10 dollars to a project because we were able to get a federal grant. We were able to get you know, a medical affairs project grant from say a pharma company or something like that. And so turning this wheel of using a little bit of local philanthropy, a little bit of our own local investment from the hospital produces those gains. We show it in live clinical care that we're able to make a difference with these technologies, create that evidence base to much more quickly move the whole healthcare system, not just London, but all of Ontario, and then for some of our work, all of Canada, to get onto this, get onto these new technologies and start making that a clinical mainstream. So in this way, it's something that starts off as a modest project here locally in London with a little bit of philanthropy support. ends up taking an idea that may have languished or not gone anywhere to clinical mainstream in a remarkably quick period of time. We have examples of less than a year where that entire cycle has been completed. That's what we're getting good at. That's the vision of the Verspeeten Clinical Genome Center, is to very rapidly and nimbly bring all these new advances to patient care every day. And I'll turn it to great colleagues who will be able to share a bit more details about what that means clinically and what that means, what that looks like in the lab as well.
Thank you so much, Mike, for sharing a bit more about the vision and how things work collectively. Our next presenter this afternoon will be Dr. Glenn Bauman. Dr. Bauman is a radiation oncologist specializing in genitourinary and central nervous system malignancies. He is also the professor and past chair chief of the Department of Oncology and is an associate scientist at Lawson Health Research Institute. Dr. Bauman's primary areas of clinical practice are in genitourinary and central nervous system tumors, and his areas of research interest include multimodality image-guided radiotherapy and cancer imaging. Dr. Bauman is going to be sharing a case study with us this afternoon, and he is broadcasting from his office.
Thanks very much. So I think we've heard a lot about the technology and the wonderful work that's been done to establish the ability to interrogate the genome and the molecular basis of different diseases through many different capacities within the Genome Center. And this is just a case to sort of illustrate what sort of difference it can make to have this extra knowledge. So this is a person that I was involved in the care of earlier this year, young man presenting with a short history of headaches and nausea and vomiting. And with that sort of history, imaging is show up in emergency room, and typically imaging of the head might be done to rule out something going on in the head, like a bleed or a mass or so. And so this gentleman had a CT scan, and then what's illustrated here is an MRI scan. So this is a cross section through the brain. So this is looking at the brain from below, sort of a section through the horizontal section through the brain. This dark area are the ventricles. So these are normal structures, fluid-filled structures in the middle of the brain. This is the brain substance itself, but this is abnormal. This is not supposed to be there. This is an MRI scan acquired after a dye was injected in the vein, which helps light up blood vessels in the tumor. And you can see this area of the tumor lights up a bit, with showing there's vascular or blood vessel supply to this tumor. And then there's a central area which is dark, which is likely fluid or dead tissue. But you can see how this is expanding and distorting the brain. And the ventricles on the right side of the brain here, this is looking up from the bottom, so this is the right side, have basically been obliterated or distorted and look very different than what should the left side ventricle looks like. And even the left side has been compressed. So the next step in managing a person like this is to do surgery. And surgery has a couple of purposes. One is to try and remove the tumor and relieve the symptoms from the tumor, and ideally get the tumor out completely, because this looks like most likely like a primary brain tumor as opposed to an infection or something. And then in the process of removing a mass like this, you get tissue, which you can then use to, through the pathology department, you can examine this tissue to just try and determine what exactly is this disease process going on. So this person had their tumor submitted to the pathology lab, and they did their usual stains and looked at it down the microscope. And this is what they saw. They saw this very uniform collection of blue cells, which means they have a high concentration of DNA, very monotonous looking. And this has a term they call it a small blue round cell tumor. And the difficulty is that this pattern under the microscope can be associated with quite a number of different types of tumor types, both tumors that arise within the brain, but also spread to the brain. So in order to understand what type of tumor this might be, pathologists have a range of other chemicals they can put on the tissue that change different colors depending on the protein composition or other composition of the tissue. And then that can give you clues as to what type of tumor this might be. So you can see this is the list of stains that they examined on this tumor. And these are all the results. So this is a tremendous amount of work in the pathology department to do all these stains. And the upshot of this was that the pattern of these stains really still did not point to a specific diagnosis. So we are still left with an embryo, it's called an embryonal CNS tumor, not otherwise specified. So in the absence of further information, we have to make treatment decisions. And we know that tumors like this can behave aggressively. They can come back in other parts of the central nervous system. They can spread through the spinal fluid and show up in other parts of the brain and spine. And so in the absence of additional information, we would typically kind of take a very aggressive approach and offer a person like this a combination of radiation and chemotherapy, where the radiation is directed basically to the whole spinal fluid space. So you can see here from a side view, this person's head and then their spinal cord. And then this is just showing all of the region we would irradiate. And then we would focus additional radiation where the tumor was originally taken out. But as you can imagine, this blue line here is radiation dose that, because of the way we deliver the radiation can reach other parts of the body. So now you're irradiating a large part of the body and potentially causing side effects, and also causing side effects because you're irradiating the whole brain, which can impair people's memory and thinking functions. So this type of treatment has a lot of side effects associated with it, and our results in the past with this type of shotgun treatment, were not all that great, because we probably have a whole bunch of different types of tumors mixed up, some that would respond well to this combination and some that wouldn't. So we started down the process of getting ready to do this treatment, but then we sought out to get other information about this tumor, and so we took some of the tumor and sent it off for molecular characterization. We tried to do the sort of genome sequencing to identify the individual genetic changes, but unfortunately we did not have sufficient material for that. But there was sufficient tissue to do the methylation pattern. And as was mentioned before, the methylation represents chemical changes to our DNA that affect how DNA is folded or compressed in our chromosomes, but also at the gene level, how genes are either turned on or off, and then resulting in different proteins being expressed, which then drive disease processes. And what's exciting about this type of methylation work is we heard about the ability to characterize different syndromes through the EpiSign project. Well, there's similar work being used within brain tumor to characterize brain tumors according to their methylation patterns. And this is a similar type of multidimensional scaling diagram, which shows that using methylation patterns on brain tumor, you can see that you have the similar clustering. So in this area, we have individuals with an aggressive type of brain tumor called glioblastoma have all similar type of clustering. Up here are pituitary tumors, very different cluster, far away from that type of tumor. Over here, we've got ependymomas. And so you can see how if you had an unknown tumor, which didn't have a typical appearance under the microscope, you might be able to see where it clusters and figure out who its neighbors are, and then you understand a bit better what type of tumor they might have. So in this paper, and this was originally described by a group out of Heidelberg, and in their paper, they had a number of cases that are very similar to the case that we are looking after, this CNS embryonal tumor not otherwise specified. So this was what they called it under the microscope, just by looking at it with the eye. And then you can see when they did the methylation pattern, these gray lines connect to where these tumors actually showed up as methylation. According to the methylation pattern, a number of them ended up looking like these glioblastomas, but there was quite a variety of other tumors. So, so this is the tumor we're dealing with. And the methylation pattern we saw actually clustered in a very uncommon tumor called Ewing sarcoma, which is a tumor that typically appears outside the brain in the muscles or the skeleton, and it rarely occurs within the brain itself. And Ewing sarcoma has a very characteristic genetic change. It has this fusion of two different parts of two different chromosomes that generate an abnormal protein, which then turns on a whole bunch of other processes that drive the tumor. And if we were able to do genomic sequencing, we would have been able to most likely pick up this fusion change. But in the absence of that, we had the next best thing, which is we were able to detect the methylation changes that this fusion protein generates, which made the diagnosis. And so now the revised plan was to proceed, instead of right to radiation, this Ewing sarcoma is a tumor that's more sensitive to chemotherapy. So this person is receiving chemotherapy first, and then we will come in and we will treat with just very focused radiation. So now you can see we're only treating a much smaller part of the brain, and less of the brain is being irradiated. We're not irradiating the spine, and the outlook for this person should be better. This tumor tends to be more sensitive to our treatments. We can tailor the chemotherapy drugs to drugs we know work better in Ewing sarcoma. And because we're treating only a small area in the brain and not the whole brain and spine, there are going to be fewer long-term side effects. So, so one of the projects within the Verspeeten Genome Center is we are developing this pipeline similar to the EpiSign pipeline. In fact, in some cases, modifying the EpiSign pipeline to do this type of methylation analysis and be able to do this type of methylation classification of CNS tumors routinely for our patients, which I think will be a huge advance and can be implemented clinically very, very quickly. So thank you for allowing me to present this case, and I'll pass it back to Pam.
Okay, thank you very much, Dr. Bauman, for sharing that case study. Our next presenter this afternoon will be Dr. Stephen Welch. Dr. Welch has been a staff medical oncologist at the London Regional Cancer Program since 2007. He holds the rank of associate professor and is the current chair of the Division of Medical Oncology. Dr. Welch has keen interest in experimental drug therapies in cancer and use of personalized medicine strategies in oncology through molecular profiling. He is one of the clinical leads for the Verspeeten Clinical Genome Center. Dr. Welch will be speaking about clinical trials, and he is also broadcasting from his office.
Good afternoon, everyone. Thank you very much for the introduction, Pam. And I'm speaking to you from my office. The background is blurred because I don't have the view that Dr. Sadikovic has from the 10th floor. I've been asked to share my thoughts on the clinical impact of genomics for cancer patients and describe some of the clinical trials and clinical research that we are conducting thanks to the involvement of the Verspeeten Clinical Genome Center. So before I do that, 3 things I want to share with everyone as a cancer treater about cancer. Number 1, cancer really sucks. Okay, I don't think most everybody could have told me that. I didn't need to be 15 years as a medical oncologist to know that cancer is a devastating disease. It definitely needs better treatment. By the time we see advanced cancer, for most advanced cancers, we are in a position of buying some time and hoping to improve quality of life for a period of time. We need better treatments, full stop. Second thing, cancer is really complicated. And it is very frustrating. It outsmarts a lot of what we throw at it. What works for one patient doesn't work for another patient. What works for one patient at the start very rarely continues to work, and then we need to keep looking for other options. But the third thing, and this is really what I want to get to in talking about the impact of genomics, every cancer is different. So even the more common cancers, lung cancer, breast cancer, colon cancer, they are not all the same. They are as different as everybody's fingerprint. I truly believe that the answer is in the genes. It's in the DNA. What is making this cancer tick? And I think that Dr. Cador, made reference to a sort of an analogy of looking under the hood of the car to see what is driving these individual cancers. And getting that knowledge is powerful. And this approach is increasing, worldwide, of not necessarily defining the cancer. This is a breast cancer, a lung cancer, a colon cancer. It's describing the cancer based on what is driving it. What are the mutations within the cancer that are driving its growth? And maybe that knowledge will help us to develop novel therapies specific to that particular mutation. So that is the type of technology that it is being used in standard practice in some situations. So take lung cancer, for example, and lung cancer is kind of it's the most common cancer there is, but we know now that we didn't know 15 years ago, but we know now that we need to construct our treatments based on the genetic information we can get from the tumor. If a lung cancer has a mutation in a gene called EGFR, it's the wrong thing to do to give that person chemotherapy. They should get a targeted drug against the EGFR protein, and then there are better outcomes. And in lung cancer, there are many other drugs like that targeting specific molecules. So in lung cancer, this type of technology is accepted, it's more standard of care, it's funded, it's available. But the same type of rationale could really apply to multiple cancers. And because there are rarer cancers that we don't have as much information as we have for lung cancer, this is all sort of a work in progress. And rather than waiting for years and years for this, for data to come in internationally to build a data set, what we want to do with the Verspeeten Clinical Genome Center is to avail patients, as many patients as we can, of this genomic information, the mutation specific to their gene, in the hopes that we can find a novel targeted therapy for them. So I did want to give one example. I should have put some slides together for this, but I can just share with you in brief how impactful this type of information can be. And I'm going to share a story similar to the one that Dr. Bauman gave of a young person with a fairly rare cancer. This was a young woman that I met earlier this year that presented to medical attention with abdominal pain and bloating, and from all appearances, it looked like an ovarian cancer that had spread into the abdominal cavity. But what was strange about this is that this type of cancer shouldn't affect someone who's 17 years old, and she was 17 years old. And so an advanced cancer, by the time it was found, the initial diagnostics were really pretty challenging to figure out exactly what kind of cancer it was. She required a biopsy and then a sort of mini surgery to get enough tissue. And we made the diagnosis of malignant peritoneal mesothelioma. Say that 5 times fast. Malignant peritoneal mesothelioma. Very rare cancer. In 15 years, I've seen 5 of them. And the standard approaches are sort of traditional chemotherapy, which is kind of like a shotgun approach. It didn't really work very well. It was hard for her to handle it. But early on, because she had a rare tumor, we enrolled her in a study that availed her of molecular profiling information, genomic information. And what we found, quite surprisingly, was a novel fusion molecule, so a gene abnormality in the gene called ALK, or anaplastic lymphoma kinase. And there is a targeted drug against ALK called crizotinib, which we were able to get for her. And since starting this medication, she has done so much better. And she's only about 3 months into this therapy, but this is making a world of difference for her. Now, I'm sure that this story is still in evolution. We're probably going to need to adjust and change because, as I said to you, point number 2 was cancer is so complicated. And so we may need to rely on this type of information again. But that speaks to, in certain cases, how important this information can be. So I think that this information, the genomic information that we can get, unfortunately, it doesn't help every patient that we evaluate. So certainly we have had, over the last 5 years, a good amount of experience. We initially conducted a research study called Octane, which was a cooperation between 5 centers in Ontario, funded by the Ontario Institute of Cancer Research. And we were able to profile through grant funding that was pretty close to $1 million of grant funding from the OICR. We were able to profile about 600 patients from London. Unfortunately, most patients, this information didn't really help them. But through this work and continued work that we're doing in this space, we've been able to enter about 50 patients onto novel targeted agent studies. One of those studies is called the Capture study that we're collaborating with the Canadian Clinical Trials Group on. Many of the patients that we have treated on that protocol have done amazingly well on targeted therapies where all other therapies were not helping them. So as I said, not everybody that we do this testing in is their benefit, but there are certainly an important minority of patients that do benefit. And what we're delivering for the majority of patients is hope for better, that through attempts to outsmart this cancer, learn more about the cancer so that we can come up with a personalized approach, a targeted therapy approach that we end up getting to better. So certainly, I think a main focus of the study that we are conducting now through the Verspeeten Clinical Genome Center is a study called POWER, that stands for Precision Oncology at Western. This is a study where, through enrollment in this study, we are able to deliver on molecular information for as many patients as we can through through donor funds that have been able to support this endeavor. And all of this research work does take a lot of coordination, a lot of effort in terms of collecting the information. We're really keen on not just delivering this information, but studying how important this is and demonstrating the clinical utility of this type of information for all the patients that we see. So we have a fantastic research coordinator in Leanne Pickard, who has been hired through the Verspeeten Clinical Genome Center that is coordinating a lot of that effort for us. And without that, we would be really struggling. So I'm very much looking forward to more innovations to come. I think that what you have heard already today about epigenetics is something that we hope we can apply through more discovery work in solid tumor cancers outside of CNS, but certainly I think that has we're very excited to see where we can apply these as novel biomarkers in cancer too. So with that, I'll hand it back to Pam. Thanks everyone for your attention.
And thank you very much, Dr. Welch, for that information around clinical trials. Quick reminder, as we're nearing the end, if you do have a question, please feel free to write it in the chat box at the bottom. We'll be happy to answer it at the end of all the presentations. So next up, we have Jennifer Masters. Jennifer will be our last presentation for the day, last but certainly not least. And Jennifer is the Director of Molecular Diagnostics, Partnerships and Innovation, including the Verspeeten Clinical Genome Center at the Pathology and Laboratory Medicine Department. Jennifer leads the clinical and translational research operations while supporting pathology and laboratory medicine with executive level relationships within the hospital and community healthcare system, and oversees partnership developments across pathology and laboratory medicine's public and commercial sector pursuits. Jennifer is working to build an environment where laboratory diagnostic innovation can flourish and accelerate. Jennifer will be sharing with us the Verspeeten Clinical Genome Center program overview and the impact of donor funding. Jennifer is also broadcasting from her office.
Good afternoon, everyone. Thank you, Pam, for the introduction. I will share my screen as well as we start to talk a little bit more about the Verspeeten Clinical Genome Center and the program of molecular diagnostics. So I'm really excited to be speaking to all of you this evening. This is my first presentation as the new director overseeing both the Verspeeten Clinical Genome Center and the Molecular Diagnostics Department or program. And I'm excited about this because it really shares the vision that we have of integrating our research and our clinical teams. And I wanted to go through a little bit about who we are and why this actually brings so much value to the outcomes that Dr. Welch and Dr. Bauman have shared with you with patients. It really is what we need to be doing to be able to drive research and clinical impact forward. So I have a research background. I did my research, my master's at U of T. And the one thing that really challenged me as I was moving through my program was the fact that I didn't see the impact that I was making on patients immediately. It was a very long road. So it was very basic research, and it would be a long road before I could actually impact patient care. And so I'm excited about what we're doing at the Verspeeten Clinical Genome Center, specifically because we're taking innovative research and we're actually implementing it almost immediately on our patients and improving their outcomes and care. So who are we? We're a team of 46 clinical staff. That includes our medical laboratory technologists, some of whom you saw as we were touring through the lab. We have medical lab assistants, our genome analysts, bioinformaticists, and of course our administrative support. We do have 7 research staff, which includes our PhDs and our master's students, as well as some postdocs, 2 research coordinators, Haley, whom you've met, and as well a bioinformaticist, Aaron, who is a new recent recruit to the team. And then as well, we have 5 clinical professionals. So we have a large team of scientists and staff that actually are supporting all of the work that we do. And I wanted to share with you some of the developments that we've accomplished over the last number of years. So since the fiscal year 2015, we've actually increased our volume significantly in terms of the number of genetic tests or the number of patients that we impact through the clinical laboratory. You'll see that within F21, we did have a slight drop, of course, related to the pandemic because some of our clinics were a little slower to be able to see patients. However, this year we're on pace to exceed over 12,000 molecular tests in this year. And so we're continuing to grow and to expand not only the menu of testing that we do, but also the number of tests and the number of patients that we impact. Sorry. And so the other piece of that is the number of genes that we actually are detecting. And so if you look at where we started, and a lot of that was with breast cancer testing, where we were testing 2 genes, and we are now growing rapidly and expanding our number of genes tested annually. And I bring this forward because it just really highlights how much that interaction between our innovation and our research team with the Verspeeten Clinical Genome Center, what that impact actually has. And so you can see that we're growing almost exponentially right now with the number of genes that we add to our testing menu. So because of all of that innovations, we have now been recruited as the only epilepsy genetic testing site in Ontario. We are the only site to do mitochondrial genetic testing in Ontario. We are the only site to do Charcot-Marie-Tooth genetic testing in Ontario. But the first don't stop there. We're actually the first center in North America to be utilizing next generation sequencing as a first tier test for myeloid cancer. We're also the first center to be offering NGS as a panel for somatic testing, for somatic cancer testing. And we're the only center globally that's currently offering clinical epigenetic testing. And as some of the speakers before me actually were talking, they talked a lot about the impact of epigenetic tests in oncology. And we do know that that is a place where we're going to continue to grow. But I want to highlight that we're really only touching the tip of the iceberg with this. There's so much more that we need to learn. And really the benefit of working with donors such as yourself is the fact that we can actually recruit scientists and researchers, which actually allows us to continue to move forward our innovations, continue to bring forward global and international firsts. Look at testing patients. So all of our donor dollars actually go to recruiting scientists and researchers, as well as providing the funds necessary for genetic testing. Now, genetic testing is not an inexpensive test to perform. And you saw all of the equipment that we have across all the different sections of our lab. And I will highlight that walking through the laboratory, you saw how broken up a lot of our spaces are. And the space was really designed for the purpose at the time. We are moving in such a quick direction. We're looking to have to renovate our space as well, because we are expanding so quickly and we need to be able to find additional space for all of these researchers and clinical staff, that our expansion is requiring a new space. And of course, technology that's associated with that is also critical. The testing that we do, the funds that we receive from our donors goes 100% to providing a testing that is not currently funded for by the province. And so with providing these tests to our patients that wouldn't normally be able to receive this deep genomic testing, we're actually able to provide the evidence that drives change in healthcare. So we've seen that with the hereditary cancer panel that we're currently offering for our patients and families that have hereditary cancer running through their family. We have expanded the gene panel by up to 137 genes. And from there, we're also expanding our panel for somatic genetic testing. And it's with this evidence that the province has decided to start to fund some of this testing, but there's so much more to do. As Dr. Welch suggested, cancer is very complicated. And so we need to do more, we need to dive deeper, and we need to understand cancer and get ahead of it before it can continue to outsmart us. And finally, as Dr. Cador mentioned, we actually are able to take the funding dollars from you as donors and multiply it. The impact becomes so significant that when we actually work with our clinicians, and we work with our pharmaceutical partners, and we work with government officials, we can actually do multiple times more of what we're currently doing. So I'm hoping that the next time that I actually have the opportunity to speak to all of you, what we'll get to do is I'll get to show you a curve that is, again, even more exponential, and the impact that we're having on our patients across Canada is going to be, across Ontario and Canada, is going to be significant. So with that, I do want to thank Archie and Irene Verspeeten. You're the reason why we're able to do what we do, and your initial donation is allowing us to do so much more, and it's really been able to kickstart that vision of us being one in the same. So pulling together the research and the clinical laboratory and doing wonderful things with all of that. And again, thank you to all of our donors. We can't do this without you, and we really appreciate you being here this evening. With that, I'll pass it back on to Jessica, and we'll be able to start taking some of your questions.
What a wonderful presentation. Thank you so much, Jennifer. That was lovely. And thank you for sharing that with us. I sure learned a lot. So now we'll be moving on to the question and answer portion of our tour. And just to inform you, I'm Jessica Da Silva. I am a development officer here at LHSF. And I'm just going to let Jennifer close off this portion. Wonderful. And I'd like to welcome back Dr. Beckham Sadikovic to help me lead this question and answer period. So thank you.
Thank you, Jessica. Thanks everyone for submitting the questions. Sorry, please go ahead.
No, it's all good. I like the enthusiasm. I'm excited to get them answered as well. So our very first question is, how many breast cancer genes have been identified?
That's good and a setup question, I think, because breast cancer is very important actually for, patients as much as it is for our lab and a trajectory of our lab. So historically, the first test for breast cancer genetics has been BRCA1 and BRCA2 gene. Those tests have been around for about 30 years or so. And in our lab, we're one of the labs that has been doing this type of targeted testing for a very long time. Of course, research and innovation in the space and the field has resulted in discovering additional genes. At this time, there are well over 24 genes that have now been with good level of certainty associated with breast cancer. What that means is that if you carry a mutation in one of these other genes, in addition to BRCA1 and 2, you are predisposed to developing what we call hereditary or familial breast cancer. The problem, though, is that we knew that information for quite a long time, and the additional testing for BRCA1 and 2 hadn't been available. So one of the first tests, actually, as a matter of fact, the first test that when I joined the lab here back 7 years ago that we developed was the expanded hereditary cancer test that included hereditary breast cancer. And with that, we went to the Ministry of Health and said, well, listen, we'd like to be able to do testing for these additional genes because they make difference to our families. At the time, we were at the forefront of this type of testing in the province, and we'd received the license to do this testing, and lo and behold, we very quickly became the provincial reference center for hereditary breast cancer testing, to a point that all of the major centers in the province, including Princess Margaret hospitals and others across the province, have been sending us testing. And fast forward 3, 4, 5 years, our initial advancement in research on this technology and ultimate implementation at clinical setting in this space of the Verspeeten Clinical Genome Center, before even it was Verspeeten Clinical Genome Center, has influenced the government, the Ministry of Health and Cancer Care Ontario, to a point where, as of about 6 months ago, this has now become a standard. So all of the laboratories and all of the hospitals in the province of Ontario that are currently doing hereditary cancer testing are mandated to do broader screening, ultimately resulting in diagnosis of hereditary breast cancer in larger proportion of our families. And of course, this is extremely important because it doesn't just make a difference for treatment of a breast cancer for that particular patient, but it allows us to test family members and prevent advanced cancers from happening in the first place. So it is a kind of a loaded answer to a simple question, but it does really highlight how innovation in this space impacts ultimately clinical care, and how investment in innovation ultimately influences the policy and allows us to change how we treat patients in our healthcare system.
Wonderful. Thank you so much. That was super enlightening. So the second question is the BRCA1 and 2 detected this way?
Yes, so BRCA1 and 2 is certainly detected this way. As I mentioned, historically, we used to do targeted assays for genetic testing for each one of those 2 genes. It was very labor intensive and very costly. With development of a next generation sequencing based technology, a custom technology that we developed in our lab here, we were able to, at very large scale, assess all of the other genes simultaneously at the same time, while at the same time simultaneously actually decreasing the cost of testing to a lesser point than it was for single gene testing, which was an amazing improvement, ultimately resulting in increased access to care. So yes, definitely BRCA1 and 2 are being tested simultaneously.
Great, thank you so much. And then question 3 is the EpiSign a commercial product of the center? And I also want to add to that, if that's okay, that during the moment of the tour, when you were speaking about epigenetics, it cut off for just a little bit. And you were noting that we were first in Canada for something, but I don't think any of us got to hear.
Okay, cool. Well, I'm going to give a part of the answer to that question. I'm going to ask my colleague, Mike Cador, to give you a bit more of an in-depth answer to this. But apologies again for that being cut off. Yes, certainly we are the first lab in Canada, actually internationally in the world, to take this concept of epigenetics, which has been in a research realm for many numbers of years, and translate it into a clinical test. Mike talked a little bit about why it's important to integrate research and clinical care within the same space. And that ultimately allowed us to fast forward a lot of this innovation and apply it into clinical management, development of this technology. So not only are we first in Canada, we're first in the world to use epigenomic or epigenetic profiles of DNA methylation to diagnose patients. Our initial goal was to focus on hereditary genetic disorders, and we're now expanding into cancer and other space. Now, we have partnered with labs around the world that are doing clinical testing to offer this technology as part of clinical care. And Mike, maybe you want to elaborate a little bit about how we think about these tests in the context of commercialization.
Yeah, sure. Thanks, Buck. In the innovation flywheel, the business of academic medicine that I was referencing, at the late stages of that flywheel, when we have technologies, tests, which are entering into the mainstream, our first place to try to sustain that is with our funding agencies and in the public healthcare systems like ours. That would be the ministries of health. In addition, we effectively commercialize live by offering the testing to other hospitals within our country and beyond. So they pay to have that testing offered for their patients. Sometimes that's in other markets where there's a single payer models like ours, other provinces, European countries. Sometimes it's insurance pays, like offering our testing to Americans. And we've got examples of all of those out of work that was initially seeded and supported through philanthropy here in London. EpiSign is, oh, sorry, I've got a page coming in the background here, so we can get that off. EpiSign is probably one of the bigger breakthrough examples where the kind of interest, potential, and if I can throw the kind of marketing phrases, the market size and our potential for market share is quite significant. And so we're going to continue to be looking for ways to sell and offer that clinically to partners, potentially even considering formally spinning out technologies like this so that, again, we can continue to drive those returns at an even larger scale and even faster pace back into the very same work of advancing patient care.
Thank you, Mike.
Thank you so much.
Back to you, Jessica.
Yes, that was great. Thank you. So our fourth question now is, I learned that getting medicine/treatment into the brain is difficult because the brain is well protected by a blood barrier. Has this changed, and do we have new methods of treating diseases in the brain?
Well, we do have an expert to answer that question. So, Glenn, are you still on, Dr. Bauman?
Yep, I'm here. So, yeah, I would say that in general, the brain is designed to keep toxic substances out of it, and that's what the blood-brain barrier does. That said, we do have existing drugs which have the ability to penetrate through the blood-brain barrier, and these include existing chemotherapy drugs, but also some of the newer targeted drugs that are designed to attack specific molecular changes. So this is definitely an area of development. There's no question that it's a challenge, and for some of the common cancers that can spread to the brain, such as breast cancer, we really do need new drugs that have the ability to get into the brain that can treat specific subtypes of these cancers. Outside of drug treatment, we have a lot of innovation in the radiation area, being able to use very targeted radiation to treat precisely cancer when it is spread to the brain. That can be a useful tool for some folks. And of course, we're always innovating with surgical techniques too. So it, tumor or cancer within the brain continues to be a therapeutic challenge. We do need new drug treatments to be able to deal with this. And in designing these new drugs, having the molecular information about these tumors is extremely helpful. Another quick example, there are certain types of benign brain tumors that happen within the brain, which can be problematic because they can grow around the nerves to the eyes and such and cause real problems. But a number of these tumors actually have a specific mutation in them called a BRAF mutation, which is commonly found in melanoma. And these tumors actually respond well to the same type of drugs that you use to treat melanoma. So we can actually and they get into the brain as well. So we can repurpose drugs if we know what the molecular makeup of these tumors are. So definitely a challenge, but an area where we are making progress.
Wonderful. Thank you so much for answering that. That was great. And then our fifth question is, does targeted therapy evaluations in its current form lend themselves better to stable cancers or aggressive cases?
Great question, and we do have an expert for that question as well. So Dr. Welsh would probably be well suited to give you some insight.
That's good.
It's a really good question. Saw it in the chat there, and I've been spending the last few minutes trying to come up with an answer. I'll be honest with you. I think that I've seen targeted therapy work well in both situations. Patients that have fairly slow growing yet metastatic disease, I've seen patients improve with targeted therapy there. I've also seen a more, aggressive, I think that, Dr. Bauman spoke to the BRAF mutated cancers, and those can be very aggressive, and they can also shrink very quickly with targeted therapies towards BRAF. So, I guess the answer is depends. It depends on the situation, but I can't say that I would shy away from a targeted therapy approach if it was very aggressive or where it was very stable. I think it really depends on the cancer situation and the target that we're talking about.
That's a great answer. Thank you, Dr. Welsh, for sharing that with us. And our last question that's appeared in the chat is, has this technology aided in the treatment of COVID?
That's a very good question. And I should just start by saying that, so DNA sequence analysis is in the crux of diagnosis of COVID. When you hear about PCR testing, it means polymerase chain reaction, which is done on a DNA molecule. In this case, it's a molecule of the virus, the COVID virus. So for the diagnosis of COVID, I should say that our lab is not directly involved, but our sister lab that's on our 10th floor, microbiology virology, has early on in the COVID response had to implement and scale up sequencing-based, DNA sequencing-based analysis to enable diagnosis of patients for COVID. Early response, I should say. I'm very proud of our group as well, because we've been able to land a good proportion of the staff that's currently in Verspeeten Clinical Genome Center to help set up and scale up and escalate the testing for the microbiology lab. And I'm wondering whether or not maybe Jen Masters would want to just talk a little bit about, I know she was quite involved in setup of our regional response and capacity for the testing of COVID. I don't want to necessarily talk about whether or not it's played a lot of role in treatment of COVID. We know that DNA sequence has been used to identify different types of variants, and we know that certain variants of COVID respond to certain types of changes in therapy, but that's still evolving. But I'm wondering, Jen, do you want to talk a little bit about what's been done from the sequencing, DNA sequencing standpoint escalation for our region here for COVID response?
Yeah, sure. So, I mean, I think we're really excited as to what our lab has been able to do from a provincial public health perspective. We have tested over 1 million patients for COVID, and we ramped up our operations from having a capacity of what we would normally do of about 30 tests a year for influenza up to a capacity of 10,000 COVID tests a day. And so we're certainly really proud of everything that we've done in terms of not only supporting our regional hospitals, supporting all of the assessment centers for COVID testing, as well as expanding into areas where the hospital laboratory has never tested before. So supporting our long-term care homes and doing genetic testing for our long-term care homes. So it's been a really, really long journey. As all of you can imagine, we've all been through the pandemic. But the technology that we've used is certainly very similar to what we do within the molecular lab, taking the DNA and extracting it and then analyzing it for the virus being present. We also are doing variants of concern testing, which is not necessarily a next generation sequencing. We're doing that through more of a probe analysis through PCR. But we are able to tell now whether or not you have a variant of concern. We've all been hearing about the Delta variant. And so that is something that we're able to inform public health of certain outbreaks with certain variants and able to enable the public health professionals from really mitigating outbreaks before they get out of control. So very similar technology, different specimen, but ultimately the benefit has been certainly for patient care.
Thank you, Jen. And I should add just one other thing, and that is that the unintended consequence of the scale up and escalation of genetic testing for COVID has ultimately resulted in a lot of our Ministry of Health government resources being funneled in direction. Inevitably, there is not a lot of money to go around, so other areas of care had been impacted. So our ability to do broader genomic testing and assessment and addition of new equipment and technologies that are specific for other areas like cancer, neurology, pediatrics, and so on have been impacted. So as a result, I expect within the coming years, we're going to heavily rely on the support of our local communities, in particular around creating capacity to continue to broaden access to genomics and genomic testing and care in areas outside of COVID.
Wonderful. Thank you so much for your responses. And that concludes our question and answer period. So thank you so much, Dr. Sadikovic, and everyone else for answering the questions and giving such insightful responses. It was very much appreciated. And also thank you to everybody for asking questions. That was wonderful. Thank you so much. And I would like to call back Pam Taylor, and she will do our closing remarks.
Okay, thank you, Jessica, and thank you so much, Beckham, for facilitating our Q&A this afternoon. Lots of really great questions. So I'd like to close out by first thanking all of our clinicians, researchers, staff, everybody who participated today. So Dr. Beckham Sadikovic, Haley, Jennifer, Risa Relator, Mike Levy, Dr. Mike Cador, Dr. Glenn Bauman, Dr. Stephen Welch, and Jennifer Masters. Thank you all so very much to you and your team for taking the time to share your presentations with us. We are so very fortunate to have your expertise right here in London. And to all of you, our donors and our partners, thank you, thank you, thank you. Thank you for your inspiration to invest in this life-saving work. Everything we've seen and heard today would not be possible without you. So thank you. We hope you've enjoyed what you've heard. We hope you've got some great things to take away. We are so grateful that you have joined us to see firsthand the impact that you are making. This concludes our tour, but I would like to wish everybody a happy Thanksgiving and a happy, safe, and healthy fall season. Thank you all again so much. Bye-bye.